Welcome to the second session in this morning's meeting entitled Theranostics. Before I hand over to the speakers, just a brief introduction. I'm Allison Jeynes-Ellis. I'm Medical Innovation Director at the ABPI. And my focus really in the role is about creating a more positive environment across the UK in terms of research and development, maximising on that investment and making sure we can attract more investment into the UK. I think Scotland's already been leading the way, we've seen in terms of basic science and translational work, as well as further down the line in clinical trials. I think pharma, when they see start-up days of around 15 days median, are very, very impressed about what actually can be delivered in Scotland, and certainly more and more trials are being placed here, as well as obviously around informatics. So I think there's an awful lot that Scotland can actually bring to this area. But as we embark on personalised medicines, it's very clear that Theranostics are going to play an increasingly important role in the new model of clinical development. And actually, to be successful in that, we're going to have to look at a very different model, more collaboration and partnerships. And that very much leads me on to our first speaker, who's Darren Hodgson, who's going to be talking about the theranostic development in oncology, what it is, when, how and why is this different to good drug development practice. Darren's very well placed to give this presentation. He is Bionomics Advisor in the clinical biomarker group at AstraZeneca, and as you'll see from his biography, he's actually worked not only in academia, but across the life sciences sector. And with that, I'd very much like to welcome Darren to the podium. Thank you.
I was going to entitle this presentation, Theranostics, What's All the Fuss? But I tried to make it a little bit more credible. So we'll talk about what Theranostics actually are and why they're different. So when, how and why is theranostic development different to good drug development practice? So I'm going to open up with a question for the audience. We've got two objects, Queen Mary II, that's the one on the left, and a new therapy there, or a new drug. I'll open it out to the audience. Any thoughts on the connection? What's the link?
Price.
Hello? Price. Yeah, development price. That's one good link. So, get it. So this is the bit I like. Usually we get some good jokes about launch speeds. We've had that one, thanks. Also about syncing sometimes, which isn't so nice. But yeah, so the price is one thing that brings these two things together. Very, very expensive development costs. These are all figures approaching $1 billion. But there's another thing that going forward is going to link these two objects. And that is that they're both, in many cases, going to need a small pilot ship or a diagnostic test to lead them safely into the harbour. Otherwise, they're going to end up floating around on the North Sea, generally unloved, unapproved, and not reimbursed. So that's what we're talking about today, this coupling of development of a therapy and a diagnostic, hence the term theranostic, or diaceutic if you come from the diagnostic industry and you want to put them the other way around. So if we look back over 100 years or so, we can look at some product promotional literature from pharmaceutical industry, and we realise some of the mistakes that we're making at present. Because we really, we don't need clinical trials and we don't need regulators because these pills recommend themselves. Similarly, we don't really need health economics, outcomes research or NICE because they're clearly worth a guinea a box. And in fact, on top of that, we don't even really need to know what disease we've got because they'll work for pretty much anything. So we'd like to think, perhaps a little bit in our molecular arrogance these days, that we've come a long way from that. But I'd like to bet that if we look back in 100 years at our product literature from today, we'll have a similar slight unease about the way we define the diseases that we're treating. Okay, so at the heart of these theranostics then is an improvement in this disease definition and the ability to prosecute predictive biomarkers. And what predictive biomarkers are hoping to do is better match the biology of our drugs with the biology of diseases. For a biomarker to be classed as predictive, it means that the risk-benefit ratio between treatment options is different depending on the result of the biomarker. So if you get result A, you're probably better off taking treatment path X if you get result B. Maybe you're better off taking treatment path Y. So clearly the ultimate value for these tests is not in our research labs, not even in clinical trials. Ultimately, it's out there in clinical practice informing treatment decisions for individual patients. But they're critical also, obviously, to clinical development to make sure we get the right patients onto the trials for our drugs. It's also not a new concept. Our precedent really for simple concepts in oncology. And think about the presence of the target, BCR-ABL and Gleevec, the levels of the target, estrogen receptor and HER2 with hormonal therapy and Herceptin, functionality of the drug target that we'll talk about in a moment with EGFR receptor mutations and epidermal growth factor tyrosine kinase inhibitors. And then slightly more sophisticated recently, the concept of a target bypass, rendering our pharmaceutical intervention irrelevant. And there we can think of the KRAS mutations and treatment with anti-EGFR monoclonal antibodies in colorectal cancer. So again, if we look back, we've not really had to coordinate this kind of thing before. So if we look here at the story of Nolvadex, tamoxifen, arguably it's a lovely 102-year story, picked up initially from Scotland, George Beatson, looking at the beneficial effects of oophorectomy through a century or more of research and development, and finally ending up with publication in the same journal 102 years later in The Lancet. And the conclusions of that paper in The Lancet were the fundamental question when assessing the proportional risk reduction that a woman can expect from a few years of adjuvant tamoxifen is whether her tumour is completely ER negative. And then it goes on to say if the tumour is shown by reliable assays. So there it is. What's the benefit-risk profile? It's linked to this biomarker, in this case the estrogen receptor, and it's essential that we have reliable assays to be able to read out on that biomarker. Okay, if we move a little bit closer in time, I'd like to put up the results of the IPAS study, or the ERESA Pan-Asia study. Not because I think it's a particularly fantastic example of drug development, but it is a nice illustration of what we mean by a predictive marker. So this was a study in first-line non-small cell lung cancer, and in these studies, these large pivotal phase 3 studies, you compare the standard of care with your new treatment paradigm. So normally on these graphs, you'd expect two curves, one for the new treatment paradigm, one for the old one. For those of you not familiar with these curves, on the y-axis we've got the probability of progression-free survival, or probability of things not getting worse, and on the x-axis we've got time from randomization or the start of the trial for that patient. And basically, more area under the curve is good. But now we've got 4 curves rather than 2. We've got to see what the risk-benefit is for the drug in not only the overall population, but in the biomarker positive or EGFR mutation positive population, and in the biomarker negative or EGFR mutation negative population. So if we move across that curve from left to right, the first group we come to, the green dotted lines there, is the mutation negative patients on the new therapy. That seems to be the worst possible place to be. If you keep moving across to the right, the next place you come to is the standard care, doublet chemotherapy for the mutation negative patients. So if you're mutation negative, it looks like you'd be better off sticking with standard of care on the basis of these results. However, if we go into the mutation positive population, key thing to notice here is that the order of these two curves is reversed. So in the mutation positive population, based on these data, it looks like your better treatment choice is to go with a new therapy. So the order of the curves has been inverted, so we have this treatment by biomarker interaction, and that is what a theranostic has to do. Help decide which treatment path you need to go down. Okay, so we'll come back to that a little bit later on. But the key thing there for now is that that was actually a retrospective analysis of that study. It wasn't a primary objective for the study, and that had a lot of consequences that we can talk about. What if we wanted to do this on the front foot? We want to coordinate the development of the test, coordinate the development of the drug, and release them both onto the market at the same time. This is what a lot of pharmaceutical companies have done, and what all pharmaceutical companies are now gearing up to do, changing their business model, changing research and development, changing clinical development in order to be able to do this. And that title, New Drug and Diagnostic Gives Cancer Patients New Hope, is deceptively simple. Under the bonnet or under the hood of that, there's quite a lot of activities to coordinate. So you can imagine the subtext for that press release is that Nostics, a diagnostics company, is announcing the availability of a new test. That test is reimbursed and regulatory approved. At the same time, a pharmaceutical company is announcing the availability of a drug, and the treatment with that drug is indicated for patients whose tumours test positive with the test that we've just talked about from the diagnostic company. Wherever they are in the world, patients will have to wait no more than 2 weeks for a clear decision on their suitability for treatment, and the test itself is also accepted by the clinical community and embedded in clinical guidelines. So there's a lot to do. There's a diagnostic company involved, a pharmaceutical company is involved, and in their label they've got a link to the diagnostic company's product. There's something there about access to the technology. If patients are not tested, they're not going to get prescribed the drug. And there's acceptance in the clinical community, not only of the drug, but also of the test. So this makes a big difference. And we've also got to take into account that traditionally these two businesses have operated in very, very different ways. So for those of us who sit present in the drug industry, we're used to working in an intellectual property-driven environment. We're used to high risks, and hopefully we get high returns. There's value-based pricing on the products and general protection from generic products. And we accept that what goes along with that are generally higher regulatory hurdles. Traditionally for the diagnostic industry, this has been a low-risk, low-return space, cost-based pricing, no generic protection, and an emphasis on making your money out of platform technologies rather than the kits that sit on them. And also traditionally there's been much lower regulatory hurdles for this space. The exception now, as we'll see later, is for therapeutic decision making in the US. Okay, so we've got to straddle those two business models, and we've got new considerations. So a pharmaceutical company is used to considering the investment at risk, the probability of success, and the return on investment to change global prescribing practice. That's fine, that's what they've always done. Now we've got a layer on top of that. We've got to consider the investment at risk, the probability of success, and the return on investment to change global diagnostic practice in order to change global prescribing practice. There might be other things as well. We might have to think about changing methods for taking and storing human biological samples in order to change diagnostic practice, in order to change prescribing practice. It's probably no coincidence, for example, in breast cancer, the three markers that get used for making treatment decisions, estrogen receptor, progesterone receptor, and HER2, are atypical in terms of their dynamic range in human tissues. They've got an enormous dynamic range in human tissues. In other words, that intrinsic biological variability is large, and we can still pick it up despite our best efforts to destroy the signal by messing around with the tissue and fixing it and leaving it in various places before we make those measurements. So we might have to consider changing the methods for taking and storing human biological samples. Similarly, we might have to consider creating global access to a new diagnostic testing platform in order to change diagnostic practice, in order to change prescribing practice. So these are non-trivial considerations for a traditional pharmaceutical company. This theme of changing diagnostic practice to change treatment practice impacts on all the functions: commercial, scientific, the design and the delivery of clinical trials, our regulatory interactions and partnerships, and also diagnostic partnerships for those pharmaceutical companies which do not have a diagnostic division, such as AstraZeneca. Okay, so the commercial stuff sounds pretty obvious, but it's key. It's going to need to be a combined drug diagnostic product profile with a robust return on investment for that combination product. And the costs for development and launch have to be commensurate with return. If we're targeting a smaller patient population, probably need to have cheaper, slimmer, sleeker development in order to make a return on that investment. Uncertainties are possibly going to remain around clinically relevant thresholds and hence final market sizes, potentially until quite late in development. Access to testing, as we've just seen, is going to be key, and all potential barriers are going to have to be addressed. Intellectual property, reimbursement, regulatory approval, simple cost of getting the testing done, the speed of testing, the geographical access to testing, and access also to diagnostic platform technologies. And lastly, new customer groups are involved. For example, in oncology, pathologists and lab staff. They need a different sales and marketing approach than that's used for the therapeutic. For the scientists in an organisation, key questions are how will defining and creating, if you like, these new patient populations increase the probability of success for the product? And this activity has two arms, inspiration and perspiration, as we probably like to call them. On the inspiration front, we've got the pure science. We're going to need to have a mechanistic rationale. Nothing new there, but a mechanistic rationale not only for the drug, but also for the biomarker interaction with the drug. We're here looking for things like association and ideally causation of sensitivity to the drug or a specific mode of action in preclinical models, and evidence that that differential sensitivity is at relevant, pharmacologically relevant concentrations. And also evidence that the biomarkers are not solely prognostic. In other words, they tell you who's going to do well irrespective of what treatment the patient gets. On the perspiration front, this is key and traditionally and not a very sexy area to work in, but it is incredibly important. We need to get better at the applied science of developing the assays for these predictive markers. They have to be measurable by robust and reproducible assays, which can be performed on relevant and available sample types, and these assays have to be transferable to CROs. So we need more than just the assays in order to be able to design clinical studies. These clinical studies have a new objective. They are there to address the value of both the drug and the biomarker in combination. And such studies can only be designed if we can set hurdles for the effect size in the subgroups defined by the new tests and the overall populations. So we need these robust tests. We also need to know how many patients are going to give us a result and the distribution of those results. The prevalence of those biomarkers is going to totally drive trial design and prospective testing needs, as we'll see in a moment. What we'd also like to have are the standard of care outcomes for the subgroups of interest. We want to check that there really is an unmet clinical need in those subgroups, and we need to know whether or not they're better or worse served by existing standards of care. And for all these reasons, tissue banks are absolutely vital at this stage. Access to representative samples and clinical outcome data from those patients. Otherwise, we cannot design the clinical trials. So if you take away anything at all from this presentation, and I hope you take away more than this, but if you take away anything at all, please take away this slide. This is really the object of the clinical development programs, is to find out where you land on this decision grid. In other words, what is the clinical utility of the drug, or what is the clinical utility of the drug and biomarker combination. The pharmaceutical companies used to working here solely in the single axis there, the horizontal axis. So we've got basically some arbitrary figures there for hazard ratio there. 1 means that you've found out that your new treatment paradigm is basically the same as the existing one. As the number gets lower, or as we move from right to left as you look at it, then that's better for the new drug. So we've now got a complete orthogonal axis to this, which is to bring in the impact of the candidate diagnostic test for the theranostic combination. And we want to ask, what's the question, what is the risk benefit for the new drug solely in the patient population defined by the diagnostic? And so clearly, if we're in the bottom right-hand corner there in red, we've not really got a case to make of taking the drug forward for anyone. It's no better than standard of care. If we're in the top left quadrant, that's a nice place to be. That means that we're getting a good risk-benefit ratio for the overall population. It's as we move across the top there from left to right where we start to see the differential risk-benefit between the overall existing definition of the patient population and the risk-benefit in the population defined by the diagnostic test that we start to get more and more into the realm of theranostics. So here we're looking at differential risk-benefit in the subpopulation versus the overall population. So we want to know where we are on this grid in clinical development. So we've got some choices about clinical study design. We could do a selected clinical trial. This would be smaller than the traditional design. Not necessarily smaller in terms of the number of patients screened, but certainly in the number of those patients enrolled. And there might be strong ethical reasons why these are the only kinds of patients you want to enroll into that trial. However, if you do this small selected study, which you might do as a proof of concept, you're never going to find out where you are on that decision grid because you don't know how good the drug is in the biomarker negative population. Another option would be to do a clinically enriched trial. So the ERESA trial I showed you is an example of that. You can enrich for a certain biomarker subpopulation using existing clinical classifications. In that particular case, you might want to look at female Asian non-smokers to enrich. You also have the option of using biomarker assays prospectively to enrich your patient populations. If your biomarker positive population is only 10% of the standard population, if you embed prospective testing into the trial, you can increase that maybe to 50% of the patients and start to decrease the error bars that you would get on finding out where you were in that subpopulation if you only recruited a few of them into your clinical trial. And the latter option may be more open to you if you have a biomarker positive population with a reasonable prevalence, say 30 to 70%, where you can recruit patients as normal into the trial and then segregate them to arms afterwards with retrospective testing. So that's the trial design, to try and find out where you are on the grid. Just designing the trial isn't enough, you've got to deliver it as well. So the clinical trials themselves look different. So the biomarker aspects of the trial have to be included in the study feasibility. So if you cannot get samples from certain centres or certain geographies, then you can't do the trial there. In oncology, this often involves consideration of a key stakeholder, a novel key stakeholder in the process. That's the pathologist. So the pathologists are the custodians of the tumour tissue, and they're the custodians of diagnosis, and they have to be engaged full-time into the clinical studies. So I said that I'd use the ERESA example as a good example of what a predictive biomarker was. But actually, if you look a bit more closely at that trial, it's not a good example of other things. So that interaction, which is potentially changing clinical practice, was based on the data from only 36% of the patients who were enrolled into that trial. So we had over 1,000 patients in the trial as a whole, and those curves were generated only from 36% of the patients. So there was issues with getting consents correct, logistics correct, and assays that were fit for the kind of samples that were routinely corrected across all those different geographies. And it's not just AstraZeneca that's had these kind of issues. If we look here at a table in the submission to the Oncology Drugs Advisory Council last year about cetuximab and KRAS testing, highlighted there in red is telling you a similar figure for the proportion of evaluable samples from patients recruited into pivotal cetuximab trials. And we've got some good numbers there, up to 70%, but we've also got some bad numbers there. Pivotal phase 3 trials, 23% of the patients enrolled in the trial ended up giving a biomarker result. So we're potentially going to want to find evidence to change clinical practice, and we've got to do better than 20% or 70% of the patients enrolled into a trial not helping us find out where we are on that decision grid. And it also leaves us open to huge problems with regulatory authorities who will immediately start to think there's some bias lurking there in the results from the trials. So it is key for those CROs and CRAs involved in delivering trials that they not only now have targets for patient recruitment, but they have targets for sample recruitment and biomarker data recruitment. And this was what we would embed now in all clinical trials. It's part of the contract. Deliver us patients and deliver us samples. So that last comment about bias in the data and not having data from the overall population brings us nicely on to the regulatory considerations for Theranostics. This at present, I think it would be fair to say, is a bit of a minefield, or on the slides I'll say it's in a state of flux worldwide. I think it's also fair to say that currently the US situation is the most stringent. The parallel process for the drug at CEDAR and for the diagnostic at CDRH. And the clinical package that you're going to have to take forward for your theranostic is going to have to satisfy both of these groups of people. And it is also now quite clear that if you don't get diagnostic approval, this is on the critical path to drug approval, and we'll look in a moment at an example where lack of diagnostic approval has stopped consideration of drug approval. So this is the kind of busy slide that everyone puts up at these kind of meetings, and it is busy, and it's I don't really want to go through it in detail, but just from a regulatory perspective, I'd like to highlight sort of two key nodes in the process, and they're in the boxes there at the bottom of this slide. So probably the first key interaction with the regulatory authorities is this so-called pre-IDE meeting, or investigational device exemption. Very confusing terms in diagnostics for those of us who took several years getting used to the acronyms in the drug industry. Then getting used to the acronyms in the diagnostics industry is just as bad. And usually when a drug and diagnostics company meet each other for the first time, there's usually about an hour of talking about things with the same acronyms that actually mean completely different. So we have to end up with a lexicon and agreed vocabulary. So the first meeting is this pre-IDE meeting, used to discuss the analytical and clinical validation involving a diagnostic company, CDRH, and pharma and CEDA. Second key part of the process is the so-called PMA, or pre-market approval, and this is effectively the application to be able to have that drug approved. Have the diagnostic test approved. And as we've seen, for a theranostic, that data is going to come from the clinical trials involving the pharmaceutical company's drug. And so this slide just shows you an example to say that this is not a theoretical consideration. This is happening, it's already happened. So here again, some excerpts from the Oncology Drugs Advisory Committee in the US. And just says there in the red box on the left that ODAC members were not asked to vote about safety and efficacy of the drug. They were asked to vote about whether or not the company should be submitting a well-characterized in vitro diagnostic for the biomarker to accompany that drug. And clearly the vote was 7-1 in favour of that outcome. So it's not theoretical, it has happened and it will happen. Hopefully not for projects I'm involved in. So this means, quite clearly, for pharmaceutical companies without diagnostic divisions, that US market access will depend on the regulatory competence of their diagnostic partners. So we've seen no diagnostic approval may lead to no drug approval. The diagnostic partner may also be heavily involved in commercializing the diagnostic test, but access to quality testing does not necessarily equate to access to the regulatory approved test. So unfortunately for the diagnostic companies, presently there is no evidence that payers will insist on using the same test that was used in pivotal trials. So this means that we've got this potentially paradoxical situation where we've got a huge stringency within the clinical trial environment, but then a potential for a sort of Wild West diagnostic testing after that from the payer's perspective. Okay, so I promised at the start that by the end you'd hopefully know what Theranostic was. I hope we've covered that. Please tell me if not. And also when, how and why is Theranostic development different to good drug development practice. And I think we've seen, just to pull out some highlights, that there's a huge onus here on strong and early basic science for candidate predictive markers. The hypothesis has to be there and embedded in the clinical trial program from an early stage. We also need applied science, fit-for-purpose biomarker assays and access to tissue banks and sample collections in order to get us ready to perform and prosecute those clinical trials. Clinical trials themselves have changed size and they've changed shape because they have to now determine the utility of the biomarker and the drug. And we need to embed biomarker delivery metrics in those clinical trials as well as patient recruitment. And also we may be skirted around this, we might come back to it in the questions of it, is that we need flexible development costs commensurate with the market sizes. If we're targeting small patient populations, we need to be able to make the case to our venture capitalists or to our boards that we can still get there and make a positive return on investment. All of which really lends us back into a nice circle to the point that both Malcolm and Allison have made, is that partnerships between all the various players in this space are increasingly important. Partnerships, yes, with academia, but also between companies, between pharmaceuticals and diagnostics companies, clinicians, patients, pathologists, regulators, and payers. And with that, I'd like to finish and just say thank you to all the collaborators and patients who've participated in trials. It's sometimes, as I came from pure science, it's sometimes very easy to get lost into the science of developing either a drug or a biomarker test. And it's always important, I think, to remember the commitment that's made by patients when they give us samples in oncology for these kind of studies. It's a non-trivial exercise, and without their participation, as well as all the rest of us, we'd be nowhere. So thank you.